I'm Ed Riley. I'm affiliated with San Diego State University. I'm a professor, distinguished professor emeritus, and now a distinguished research professor at the SDSU Foundation.
Well, thank you very much for agreeing to be part of this interview for the RSA Histories Committee. We appreciate your time. My first question for you goes back to some early work in your career where you published a series of both preclinical and clinical studies on alcohol drinking. And now you're the PI of the collaborative initiative on FASD that's investigating means to improve prevention, diagnosis, and treatment of FASD. How did your early preclinical and clinical research shape your career trajectory?
Interesting question. I was trained in animal learning, not alcohol at all. And after I got out of graduate school, I got a postdoc position at the Center for Alcohol Studies. That Carrie Randall, who was a graduate student with me at Rutgers, told me about this postdoc, and I got the postdoc. But it was with two different people, Dave Lester, who was a biochemist, did basic science, and Earl Freed, who was a clinical psychologist and at the VA hospital. So the animal lab that I worked in was at the VA hospital, but Earl was very insistent that if I was going to study this problem of alcohol use, that I needed to get onto the wards and see what alcohol abuse was really like. So he was insistent that I learn the clinical aspects of this, something I had never done in graduate school or whatever. So my postdoc really set me up for looking at the benefits of understanding the disorder I was trying to model in the animals. So it was just a great experience, learning experience, and kind of kept my interest in translational sciences and translational research, which CFAS is heavily involved in now.
So with that translational aspect in mind, can you tell us a little bit about how you moved along in your path in alcohol research?
Okay, so just started out, like I said, I was trained in animal learning and took this job at Rutgers Center Alcohol Studies and started working on the genetics of alcohol. After I was there a year, one of my former dissertation committee members took the chair at SUNY Albany and called me up a couple of weeks before the semester started and offered me a job. So I went to SUNY Albany as an assistant professor and knew a little bit about alcohol. I was selectively breeding rats for alcohol sensitivity at the time. Brought those rats with me. And then, by sheer coincidence, I was watching 60 Minutes. And I saw an episode right at the end of 60 Minutes, very brief, on fetal alcohol. So my friend Carrie Randall had been looking at fetal alcohol. I said, I saw Ken Jones and Buzz Chernoff, and they said the brain was the most affected organ with this prenatal exposure. This was 1976, 77. So I said, God, I know a little bit about alcohol and I know a little bit about behavior. I'm going to write a grant. So I wrote a grant. And as a result of that, I got invited to the first fetal alcohol working group. It was not a really great grant, $30,000 a year. But as a result of that meeting, I met enough people, found enough new information out that the grant got funded. And I had that grant for years, I think 20-something years. It was a merit award eventually. But it was just sheer luck that I got to meet the right people. I got in the right field at the right time. And so that kind of led to all the work that we did. And then, as I said, I was at SUNY Albany primarily doing animal work. And you could give alcohol to pregnant animals and always find a difference, right, between them and the controls. And I was really getting kind of bored with it. And I decided that I wanted to try to do the same stuff, but more clinically oriented. And so I saw a job at San Diego where Ken Jones was, and called him up and said, if I moved to San Diego, would you let me test kids? And I talked to Nelson Butters, who was at UCSD, and I said, would you train me how to test kids? And they both agreed. So I got offered the job and made the move, and then kind of ran an animal lab and a human lab for a number of years, and then kind of basically switched to doing all the kids' work.
All right, great. I want to go back a little bit further in time. Can you tell us a little bit about some influences in your childhood or high school that some folks had a special impact on your progression as a scientist?
Wouldn't be back in my childhood. I had to leave it to Beaver childhood. I mean, I didn't even know there was an alcohol problem in the world. I have uncles who owned bars, but none of them ever drank. Uh, a weird Irish thing, you know. They had Irish bars, but no one really drank a lot. So that wasn't it at all. It was in graduate school, and Carrie Randall had a big influence on me. She was the one who basically told me about this postdoc, because when I got out of graduate school, I didn't find a job, and I went to work in a machine shop because I know how to operate a lathe. And she called me up one day and said, "This postdoc," and so that's how I got in the alcohol thing. It was just the only job that was out there yet. And then when I moved to Albany, the people who had a big influence were Henry Begleiter and T.K. Lee. And I don't know why, but we got along. You know, they're two senior people, and they fostered my career. I mean, Henry got me involved in the RSA. You know, I think in the early 80s, he put me on the membership committee and then made me chair of the membership committee. Then I was on the finance committee, and you know, eventually wound up as president. So, but Henry and TK were very uh, big proponents and helped me a lot along the way.
So you've had a very productive career.
Yes.
Can you talk a little bit about the challenges that you faced throughout your time as a researcher?
Yeah, I mean, the first one was when I went to Albany. There was no money. You know, there was no big resource, no real faculty. It had just been evaluated for its Ph.D. program, and the evaluation was that most of the faculty shouldn't be allowed within 100 yards of a graduate student. So that's why they hired the new chair, and he was hired me. So we didn't have anything. I was just reminded that I used to go around and steal the runners from under people's chairs to make shuttle boxes, plexiglass shuttle boxes, because I couldn't afford plexiglass. So I just used the runners off of people's office chairs. I got known as the black hole at SUNY. So that was a challenge. I mean, and no one there had really ever had any NIH grants. So it was all a new experience for me, who was writing grants and being successful. And but that was about it. Other than that, once I got that one grant, I got a couple of grants, and so I had the funding. And having funding has always been a very useful tool.
So you've had some seminal manuscripts in your career. Can you maybe tell us about any eureka moments that you've experienced and how you arrived to those points?
Well, a couple of them, but I mean, one of the ones that I'm most proud of, I guess, or was really useful, was when I moved to Albany. And like I said, had to learn how to assess kids. And I found San Diego was just an academic wonderland. There were lots of people who did lots of different things, and it was a very collegial environment. So everybody was willing to help out with helping me set up this project to study fetal alcohol and behavioral effects. And so one of our graduate students, Elizabeth Sell and Terry Jernigan, went to them and said, no one's ever really imaged these kids. Why don't we do that? So we imaged, and the first person we imaged had agenesis in the corpus callosum. Second kid we did had a very thin corpus callosum, but we published a case report. That picture wound up on the cover of textbooks. So that was just, but it was because there was a great environment to do this cross or multidisciplinary type of work in San Diego. And it was different than I had ever experienced before, where I was no longer working in isolation. I could collaborate, and if I didn't know how to do something, I could find somebody who did and was willing to help.
The alcohol field in general is highly interactive and collaborative. And you have mentioned a few people already that had some influences on your research. Is there anybody else that you haven't discussed so far that's had an important influence on your research as you've progressed through your career?
Yeah, well, my graduate students have always, they've taught me a lot. So I work Sarah Matson was a student in my introductory psychology class in Albany, and got the only perfect paper I'd ever scored there. And I taught a class of 300 people, so I really didn't know who she was. But I found her, I located her, and asked her if she wanted to come work with me. She said no, and she wanted to go be a doctor. And a few years later, she came back and said, "I've changed my mind, and I'd like to work in your lab." And so she and I have had a 30-something year relationship, and she's gone on and she's got her own fantastic career in neuropsychology and fetal alcohol, and among other things. And so, it's great to have that kind of rapport and bouncing ideas back and forth. And so,
So speaking of graduate students, what would you like to see in the future of alcohol research, and what would you expect to see in the future?
in terms of graduate students,
Well, the type of research that they're going to do. What would you like to see the field move toward?
I've become and it may be somewhat unique to the fetal alcohol, is where very few of the kids really who are maybe affected by prenatal alcohol exposure get diagnosis and get involved. And all of the kids that I know, or the adults that I now know, because I've been following them for years, say that the diagnosis really helped them out. All right, it's few exceptions. They always say that, boy, once they learned what was the problems, the reason for the problems, it let them deal with those problems in a much easier fashion. So there's just a vast number of people, however, who are affected but don't get a diagnosis. There's a disparity between the number affected and the people who can diagnose and treat the disorder. So one of the things that I've got interested in was using telehealth and more technology to try to make the diagnosis easier, so you don't need to see a dysmorphology specialist. You don't need a multidisciplinary team to do this diagnosis, or to even screen for the disorder. So I'd like to see us move to increase the capacity by using technology.
So you talked a little bit about graduate students. Can you tell us your thoughts on mentorship, and if you have a philosophy surrounding mentorship?
I do. And my philosophy is really simple. If you want to come work with me, you have to be independent, you have to be creative, free thinker, be able to get outside of the box if you need to, and not need a lot of hand holding. Because I don't do that. And so students who have come to work with me who don't have those qualities, they don't stay around very long. They're gone. So when I had graduate students, I was always in my office and always in the lab. So it was an open door policy. We'd schedule meetings, but it usually just wound up when you had a problem, we'd come in and talk. And I was there all the time. So, but I'm not one to try to hold your hand through the whole process.
So for researchers at the beginning of their careers, do you have any advice?
Network, network, network. Get to know people, because the field has become so specialized that you can't do everything, and you need colleagues. And I still talk to people I was a graduate student with, Carrie Randall being one of them. I've maintained these relationships, and that's been important both in my professional life and my personal life. These are friends as well as colleagues, and I think that's important. And I mean, when I think early in my career, when my son was born, he had a lot of medical issues, and people from all disciplines called me up and said, "I know this person, this person, and this person. If you need, this medical help, let me know." I mean, everybody was on board. So I'd say the personal interactions have been just incredible in helping my career along, both professionally as well as my personal.
So for FASD researchers in general, do you have anything to ask of them for future research?
Yeah, I think we need to come together and decide on what FAS is. One of the problems we have now is that you read a paper from one lab and you're not sure how that how similar the population is to the kids that you may be studying or the individuals you may be studying. Uh, so I think we need, and NIH or NIAAA is trying to deal with this. There's currently a consensus working group to try to establish research criteria for fetal alcohol spectrum disorders. Um, I think we need to try to get a better handle on biomarkers for alcohol exposure. That's a tough question to answer. When the reason a lot of people don't get diagnosis is that you don't have the alcohol exposure history. It's hard to make the diagnosis of along the spectrum unless you have all the facial features. Um, so I think biomarkers is an important area. I think technology, the use of technology to screen, treat, intervene. You can't possibly intervene with all the people. There's just not enough therapists in the world to provide everyone who might be affected by prenatal alcohol exposure with appropriate intervention. So we need more online tools, online interventions. And Christy Petranko, I think, is a good example of a young investigator who is looking at e-health technologies in the field of intervention.
I want to talk a little bit more about interventions. FASD is preventable. How can we best educate the public on the dangers of drinking during pregnancy? And are there any new initiatives to increase public awareness?
Good question. And when I started out doing FASD research, I thought it would be a 5-year project and I'd be done. We'd have solved the problem. And that was the late 70s. And it's 50 years later, and I'm still baffled, befuddled by the number of individuals that are still, I think George said 11% are drinking during pregnancy, 4% are binge drinking. And considering that a lot of the effects of prenatal alcohol exposure are first trimester events, and 60% of women drink, why 60% of women drink, and they don't recognize they're pregnant. I think that the numbers of exposed are even larger than that. So, I think that's an issue that needs to be resolved.
Any advice for early stage investigators who are interested in outreach and influencing public policy to maybe curb the issue here?
Yeah, get involved. there are a number of committees at NIH and at AAA that have been trying to educate, get involved with the ICFAS, the Interagency Coordinating Committee. They have public meetings so anyone can attend and make your ideas known. Um, I think in terms of education, we've done a reasonable job, but we haven't hit the population that's highest risk. And I think we need to do more in that regard. We need to be in treatment facilities for alcohol use disorders. We need to be targeting high-risk women.
One last question for you. For the members of the public who may watch this video, do you have any advice that you would like to communicate to them?
Yeah, I would drink while I was pregnant. I mean, and I'm not. Every kid who has a behavioral problem doesn't have that problem because they were exposed to alcohol prenatally. But we haven't established any safe level. I doubt if we ever will because there are so many other factors that go into it that for different people that level's going to be different of how much you could get by with without having impact in the fetus. So the safest course is simply not to drink. And I run into women who have a child who has a birth defect or has a behavioral problem, and they said, "Well, I had 2 drinks before I knew I was pregnant," and they carry this around that they're the reason for. So it's just not worth the stress. If you can stop, stop, and cut down, cut down.